So good morning, everyone, to LA people, and good morning, Dr. Huber from Germany, or good evening from Germany. So this is our seminar in the statistics department, and we're greatly honored to have Dr. Wolfgang Huber to speak for us from Germany. Dr. Huber is a well-established statistical genomics expert, and his research interest lies in both computational biology and statistical computing. He has developed several very famous tools that has been widely used in the bioinformatics field. And especially, I think for those attendants from the human genetics and bioinformatics programs, you must be very familiar with his work, the DE-seq and DE-seq tool for analyzing RNA-seq data. And Dr. Huber is a founding member of Bioconductor, which is a very big repository for R packages about bioinformatics data analysis. And oh, sorry, I forgot to say that Dr. Huber is a leader in the EMBL Heidelberg. It's actually a very big research institute in Europe. And so regarding Dr. Huber's backgrounds, he has a very interdisciplinary background. He first studied physics in University of Freiburg, and then he got his PhD also in physics. And then he moved to California, actually worked here for quite a while in the industry sector. And then he started working with genomics data in the early 21st century. And then he moved back to Europe and started his research group. So today he's going to give us talks about his very interesting recent work about two methods. One is called independent hypothesis weighting, and the other is called multi-omics factor analysis. And we very much look forward to your talk. Thank you, Dr. Huber.
Thank you very much. So I'd like to thank the organizers of the statistics seminar, and in particular you, Jingy, for your kind invitation. And I'm excited about the opportunity to present some of the research here. So as mentioned, I'm based in Heidelberg, Germany. This is about 9 time zones east of you. And I work at EMBL, at the European Molecular Biology Laboratory, which is an intergovernmental research organization with 27 member states, sort of founded in the image of CERN for physics. So the idea was to have something similar for biology. And it now has sites across the continent. It's actually several different places, and it's a fairly sort of international and cosmopolitan place. And what you see here is a picture, of course, of Heidelberg, an emblem somewhere at the top right of this picture. So the missions of the institutes are fivefold. They include basic research, but also the development of technologies, of transfer of technology basically into industry and society. We provide scientific services, which the most prominent of this is the EBI, the European Bioinformatics Institute, which is one of the biggest databases for like genome sequences and genome browsers and all sorts of data types. And also that there's missions to provide training to scientists around the world. So EMBL offers opportunities for scientists from many different areas, obviously biology, but equally important and maybe even more in demand, from chemists, medics, but also physicists and mathematicians, statisticians, computer scientists, engineers, and so on. So, it basically offers career opportunities at all levels, including internships, PhDs, postdocs, PI jobs, and so on. So if you're interested, I'm also happy to talk about this. Being an international organization, the place is very cosmopolitan, and I guess immigration and visa formalities are very lightweight and easy to deal with here. So now in this talk, I've organized it into 2 essentially independent blocks. One is a bit more applied, data-driven. The other is a bit more conceptual and theoretical. I think with a wide range of applications, far beyond bioinformatics that we haven't even yet scratched the surface of. The second one. So first about multi-omics. So what do we mean by that term? So we try to characterize a set of biological samples, for instance, tumor biopsies, not just with 1 or 2 types of omics assays, but with all of them, or at least as many as possible. So for instance, you might use genome sequencing to get at the DNA mutations in a tumor, but also look at DNA methylation patterns and chromatin accessibility, a transcription, proteome, possibly protein modifications like phosphorylations, metabolome imaging, and indeed functional assays such as drug or cytokine responses of the tumor cells. when cultured, like in a cell culture and an organoid. So the aim of this is, of course, as you probably all know from this meme of the 6 blind men and the elephant, to get a comprehensive and complete overview of a complex system rather than falling into a reductionist trap. So to establish multi-omics-based precision medicine for blood cancers, I teamed up with a team of sort of biomedical scientists at the University Hospital in Heidelberg and also in Zurich, which is now a team of about 20 researchers that are working with different aspects and across 3 different sites, my lab at Amble and the hemato-oncology departments at Heidelberg and Zurich. And so we use directly the primary tumors, now over about 4,000, which under the right conditions can be stored in a freezer and basically brought back to life for cell-based assays. So these tumors are indeed viable outside the body, at least for a while, without immortalization. And we characterize them both with the full omics tool set, like exome sequencing, RNA sequencing, DNA methylation, and so on. But also we obtain interventional phenotypes by applying in parallel perturbations using a cancer drug library and or cytokines. And in this way, we can really cover the natural genetic heterogeneity of the disease. We can test multiple drugs on the same tumor in parallel and sort of compare the outcomes, something which obviously you can never do with the patient. We can also use tool compounds to probe individual pathway dependencies. And the hope is to use this to discover phenotypically important molecular subtypes to maybe find repurposing and combination opportunities. The insights do inspire new clinical trials. I mean, as you all know, clinical trials are expensive and take a long time to prepare. So it's better if you go into them already with a very good biological hypothesis understanding, and that makes the quality and the success chance of the trial much better. And I think the kind of data that I'm showing are intended to help with the design of good clinical trials. And sometimes these sort of ex vivo functional assays that we can do with patient material also have found direct application in molecular tumor boards when basically the standard therapy options for patients were exhausted. Then maybe the more adventurous ideas that you can get from applying such a whole drug panel to a tumor also have found uses, and in some cases actually with spectacular success. And we also have a more systematic prospective clinical trial where we sort of showcase the feasibility of doing these functional assays on the tumor ex vivo in real time so that they could be used more routinely in clinical care. Now, from a statistical viewpoint, multi-omics is actually a mess. I mean, if you have batch effects and incubation effects and contaminations and all of these problems in single omics platforms, this just grows exponentially when you have multiple different omic technologies. There's a problem of missing values. So even though you may design that every sample is done with 5 different technologies, of course, in practice, some things won't work out and you will have to deal with missing values. And the data types are quite heterogeneous. So they're heterogeneous. They can range from binary, I'd say, with mutations, when you maybe think about their present or absent, to continuous valued like drug responses or DNA methylation scores, to actual counts like an RNA-seq. And the statistical models have to be able to deal with all of these different data types and their associated error models. And of course, the complexity also goes with such more complex experiments. So I'll just leave this meme up here for a few seconds to appreciate the difference in style and approach between the classical single technology omics experiment and the kind of multi-omics studies that I mentioned in the previous slide. So what's a first analysis or front-end analysis that you could try to apply such a data set if somebody asks you to have a look at it? And one option, which we have recently proposed, is the MOFA method. MOFA stands for multi-omics factor analysis. And simply speaking, it's a matrix decomposition method, very similar in spirit to a single value decomposition or principal component analysis. But instead of working on a single data matrix, it takes multiple data matrices on the same set of samples. So as indicated in this graphic on the slide, the samples would correspond to the columns. So different patient biopsies, for instance. the different matrices to different, I apologize, to different omics technologies, for instance, metabolome, genome, and proteome is indicated here. And the rows of these matrices are different features like genes, metabolites, or proteins. And what the method would do, it basically finds a common decomposition of these multiple matrices into a common factor matrix Z, and only this view or data type specific matrices W. And the idea that MOFA is it can deal gracefully with missing data, so it's indicated here. Some of the columns in these Y matrices are missing, maybe because the assay didn't work for them. It can also handle different error models, and it aims at model sparsity, as I'll explain next. So the way that MOFA is implemented is through a Bayesian model for the common factor matrix Z. So the model is sort of written down again at the top left. For the common factor matrix Z, we just do standard normal prior, whereas we put a spike and slab prior on the elements of W. And then by doing so, we can basically achieve two levels of sparsity. So with theta, which controls this Bernoulli process and the sparsity. So the matrix W, basically, the matrix W, as indicated in the graphic at the bottom left, is the product of a Bernoulli variable S and a normal variable W bar. And by doing so, we can basically get a sparse model, because many of the S's will be 0. The model inference can handle missing values, and we can handle different noise models in this here. So it currently implemented are normal Bernoulli and Poisson data. The inference is done using a mean field approximation, and I come back to this. The package should be fairly easy to use. It's written in Python, and our Biconductor has examples, tutorials, and helper functions for exploring a certain model. So I recommend you have a look at it if you have such data types. I mean, I've really seen quite amazingly how this is, many people in the group and in collaborating groups use this now without having to be encouraged to do so as a first analysis tool to get an overview of a new data set and sort of start exploring what's going on in a new data set. So this is by no means the end of the line or perfect. I think there are many open ends in this model. Because of this mean field approximation and the inference, it basically gives you back a point estimate of a model fit in the end. So there's no, sorry, there's still a need for better handling of model uncertainty, identifiability issues with the factor analysis, and also model complexities and the number of components. We're also currently working on, and this is, I think, still some way to go, on how to best incorporate explicit covariates, such as, for instance, disease subtypes, so known disease subtypes, or for instance, if you now look at single cell data, you could think of the latent factors as different cell types, but then you have also different patients or maybe different treatment conditions, which you would like to sort of import using an explicit covariance matrix X. And then maybe your factors should be different for different levels of X. So how to implement such interactions between explicit covariates and latent factors, I think there's, and then of course also how to estimate such a model on real data efficiently. There's lots of open ends here. Another thing we're working on is to implement the gamma Poisson noise model, which is really needed for single cell data for fitting this. What I do want to show now in the next 5 or 6 minutes is in fact an example of how one can use this method and the richness of a multi-omics data set to find new and unanticipated new disease dimensions of certifications. In fact, this was quite surprising that this disease dimension had been overlooked before, since our disease that we're looking at here, CLL, chronic lymphocytic leukemia, is a really well-studied and quite prevalent disease that, I mean, at some level has been studied to death using omics technologies and genome sequencing and RNA sequencing and whatnot before. And still there's things to be discovered. And some of the things really only came about by doing this multi-omics analysis. So this is now our blood cancer data, and as the box at the bottom left indicates, this in a graphic, we had 217 CLL patient samples. For all of them, the clinical a panel sequencing of the mutations was done. For most of them, also we had mRNA profiles from RNA-seq, DNA methylation profiles from DNA methylation chips, and drug response assays from our ex vivo drug response screen. And this is so basically we could input these 4 matrices into the method, and what it then basically returns is this factor analysis, and then using some threshold of effect size similar to a scree plot in PCA. The model decided, or the fit decided to have seven sort of important factors in here, and their loadings are indicated in the matrix on the bottom left. So this purple-white matrix. So how to read this? The 7 factors correspond to the 7 different rows of the matrix, and the columns to the different data types. Basically, the coloring corresponds to the weight of each of the factors in the different data types. And what you see, the first 2 factors, F1 and F2, they have loadings in all data types. So basically, they're active in all of the different data types, where some of the other factors then are, for instance, more mRNA specific or also across the different data types. So what was reassuring is that the first 2 latent factors, in fact, recapitulated known important stratification markers. So without inputting IGHV status or trisomy 12 in a supervised way, the model just essentially found a 2-dimensional projection of the samples that then separates those variables almost perfectly, as you see in the scatter plot on the left. So that's reassuring, and basically tells you that that the first 2 factors sort of reproduce known biology. And then of course the question is, what about the other 5 latent factors? So we screened all of them against disease outcome, which again was a variable we didn't use for model fitting. And of them, only factor 4, but especially factor 4, was associated with clinical outcome, and I think also with lymphocyte doubling time. So that's basically the speed in which the tumor grows within the patient. And that's of course an interesting finding, and this finding was there both in the univariate analysis, but also in a multivariate analysis when we basically used all the known risk factors. Factor 4 in addition provided predictive power in this multivariate Cox regression. So that was interesting. And in fact, then a whole sort of months of additional analysis and trying to interpret what this factor 4 means followed. And this is just now summarized in this slide very quickly. That basically we could now see that this factor 4 associated with multiple layers of disease biology, like it basically measures the acquisition of genetic operations. So the higher the value of factor 4 is, the more operations the tumor is collected. In fact, it also correlates with a mitotic clock. So somehow it counts, measures the age of the tumor, and it is in terms of cell divisions that it has undergone. And it also measures the metabolic activity and sensitivity of those tumors to specific drugs like rapamycin and beta inhibitors. That was all very interesting to find. And I guess one question that now follows, obviously finding this factor was a very involved process using multi-omics and a complicated method. Could it be easier? And the key insight now is, of course, once we identify this factor for multi-omics, we can try to predict or approximate it from single omics data types. It turns out that out of our 4 available views, RNA-seq and DNA methylation chips alone are already quite good in doing the jobs. So we can basically now find profiles just based on these assays that already predict the factor 4 value for a new sample quite well. In fact, interestingly enough, also other data types don't do that job. So, but anyway, using the RNA-seq then score pattern, we could then apply our factor 4 idea to independent data sets. And what I was going to show on this slide is simply now a summary of the analysis on 4 different independent data sets, where in each case, again, this expression profile that corresponds to factor 4 seems to distinguish the disease, or certify the disease in terms of a meaningful difference in outcome. And this is true also in the multivariate analysis, where the data were available for that. So to summarize that, so basically, even though this disease had been analyzed, as I mentioned, quite extensively already using omics, this factor 4 had been overlooked. It was discovered by an unsupervised machine learning algorithm on the multi-omics data. It validates on the now independent data sets. It's nice that it can actually be captured and now measured once we know about it to a cheap single omics assay. So we're hoping that this could in fact be measured for a few dollars per sample, using, for instance, assays developed by some collaborators like Chris Oakson at OSU. And it maybe have potential use to identify patients for therapies, for instance, using targeting mTOR BET proteins. So those drugs have been discussed in this disease, but generally haven't been used because they don't work, because efficacy hasn't been uniformly good. But of course, but now if you have biomarkers for these drugs, in fact, the efficacy may be better and they may be more applicable. These are really interesting sort of developments. And sort of a general observation from this, that single and multi-view factors can be in general useful if you have multi-omics data set to distinguish technical from biological variability in multi-omics data set. So if we recall, we had found 7 factors in our factor analysis, and the other 4, so that were not 1, 2, and factors 1, 2, and 4, so the remaining ones, they were essentially exclusive to the mRNA data level. And we think that probably mostly just batch effects, or experimental artifacts of reagent aging, or somehow drifts in the protocols that were used. So this is, I think, a general idea that Emma, one of the students in the lab, has found to actually apply in multiple different application scenarios. In many cases, the factors that are common to multiple layers of biology are for more immediate biological interest, whereas the factors that are technology specific also tend to be maybe more ephemeral and more like artifacts. Of course, this is no hard law, it's more like a rule of thumb, but it can be very useful when looking at such factor decompositions. So now I'm sort of switching gears and moving into the second half of my talk. So it's a little bit of time to stretch and refresh. So I'm now going to talk about multiple testing and using covariates. So, I mean, as probably most of you know, multiple testing is basically screening for associations in high-dimensional data sets. This is something we do a lot in genomics, but I think also people, let's say, in high-energy physics, or economics and psychology, other fields, social sciences are doing this as well. They have big data sets and are screening for lots of interesting associations that they can then follow up. In biology, this is really pervasive in expression profile, obviously in GWAS, in gene or compound screens, in proteomics, in microbiomes, and I mean, you name it. Very many analysis techniques really consist of having a lot of hypotheses and then screening through them to find the interesting ones. Now, multiple testing in the statistics world has often been associated with a burden, and that is maybe related to the observation stated here, that when you perform several tests, the type 1 error goes up. So for instance, if you have a type 1 error of 5% per test and do n independent tests, the probability of no false positive result in the end is the product of these 0.95s, and that number, of course, very quickly becomes very small and approaches zero for reasonable values of n. That's the multiple testing burden. But actually, what I want to argue in the remaining part of this talk is that the multiple testing offers big opportunities. And I'd like to explain this using this XKCD comic, which really, I think, captures the situation very perfectly, very well. So here are two scientists that have a machine that detects when the sun goes supernova. And the machine has an error rate of 1/36. For some reason, that's how it works. And it sort of beeps sometimes with the type 1 error of 1/36. So now actually the data is that it does beep. The frequentist takes this data point, computes a type 1 error, which is 1/36. That's less than 5%. And therefore, they call the test outcome significant and announce that an explosion might have happened or should have happened. The Bayesian statistician would actually know a little bit about astrophysics, that the sun's mass is too small, that it'll only ever become a red giant or then a white dwarf and cool off, but then never actually explode as a supernova because it doesn't have enough gravity. So the physics knowledge about the world would inform the Bayesian about her prior, and her strong prior together with the weak data would then maybe lead to her to conclude that no nova has happened. Now, the empirical based person doesn't actually know any physics, but she's been around for a while and has observed that the machine already beeped several times before and nothing happened. There was no nova. So for her, the multiple testing, having observed the machine before many times, also gives her an opportunity to build a prior, even without knowing much physics. But she can then still use that to better analyze the data and also reject, not call a wrong result. So that's sort of the cartoon version of what I'm talking about. And I want to apply this now to real data sets or demonstrate applications to make this more concrete on two biological data sets. One of them is again coming back to our CLL studies, where we have gene expression as well as the response to a panel of drugs measured by some viability assay. And basically the data that we have here, the hypotheses that we test are like shown in this plot on the bottom left. So relative viability, each point would correspond to a tumor. On the x-axis, the viability after drug treatment. And on the y-axis, some measure of gene expression. We can now do this for all pairs of drugs and genes. So if you have 300 drugs and 10,000 genes, that gives you something like 30 million hypotheses to test. And we can indeed use DE-seq2 to run these tests on our DE-seq data, when basically the drug response is the covariate and the counts would go in. So that's one example where we can use the idea of multiple hypothesis testing is evident. As I mentioned, the number of hypotheses here is in the order of 30 million. Another one is a genome-wide association study, in this case, a histone EQTL study, where the data that we use here was produced in Stanford by Mike Snyder's lab. So we have the genome, the SNP calls for a set of individuals, in this case, 75 individuals that were sequenced, where the SNPs, the genetic variations were called. And at the same time, we had ChIP-seq data for histone marks of these individual cell lines. So basically, these are measures of the activity of the chromatin at each place in the genome, measured by a ChIP assay. And then again, we can basically look at associations between DNA mutations and these ChIP-seq signals. So if we, for instance, have 1 million SNPs that we can look at, and several thousand of these ChIP-seq peaks, we could test each pair of them, and then again would have something in the order of, I think, 11 billion or so tests that we can perform for associations. So again, a huge multiple testing problem. So to now actually crack this, let's recall the workings of p-values in terms of the two groups model, which, as far as I understand, was popularized by Story and Efron. The two groups model really is shown here in this graphic. So what you see is the distribution of p-values. So the density, this is what is shown here is the density of a p-value, and you would consider it as a mixture of two underlying distributions. So the gray underlying distribution is uniform, which would correspond to the null part of the model, and the blue density corresponds to the alternative distribution of the p-value. So this is concentrated towards the left, towards small values, as a good alternative distribution should. And the parameter pi naught corresponds to the mixture ratio between nulls and alternatives, or null and alternative. And so you have basically this parameter. So you have the uniform distributions, the pi naught parameter, and the shape of the alternative distribution, the blue. And from this, you could already, for instance, if you now call everything to the left of the red line significant, so if the red line corresponds to your p-value cutoff, then you can immediately from this model read off your false discovery rate, which is basically the ratio of the overall area to the left of the red line to the gray area, to the dark gray area, because the dark gray would correspond to your false rejections, right? Or if you do this in the local way, if you look at the value at a specific p-value, this fraction between pi naught and pi total would again be correspond to a local false discovery rate. So this is probably, I hope many people have seen this. It's just to recall the interpretation of p-values in terms of the two groups model and the shapes of the alternative and null distributions. The main point here is that there are two critical parameters, the value of pi naught and the shape of the alternative density, like of the blue density. And to make this clear, we can have variations in these parameters. So in the center, so I now have three of these plots, and the left one is the same as I had before. In the middle one, we have the same mixing ratio, the same value of pi naught, but the shape of the alternative density has changed. It's now a bit flatter, which corresponds to less powerful test. So with this flatter alternative distribution, the overall story changes. Or in the right-hand side of the in the rightmost diagram, in fact, the shape of the alternative distribution is the same, but the pi naught changed. So we now have a higher ratio of nulls. The gray area is bigger, and the blue areas are smaller. And what this indicates is that with the same p-value, so with the same red line, you can have very different false discovery rates. That's the main point to make here. So that there's no one-to-one relationship between p-value and false discovery rate, and it changes in these both in the pi naught and in the shape of the alternative distribution can change the relationship here. Now, empirically, we can already see this also often in the histogram of p-values from an actual multiple testing experiment. And I'm showing now here just actually showing some real data from one of the data sets that I showed before. The histogram of p-values, you can visually, in fact, fit the blue line, which sort of corresponds visually maybe to the nulls, whereas the hypothesis or the p-value counts above the blue line might reflect the alternative distribution. And then, for instance, if you draw a cutoff at where the red line is, the vertical red line, you can already visually estimate your false discovery rate that corresponds to that simply by dividing the dark blue area by the overall length of the first bar. So that's sort of your visual eyeballing FDR estimate, just taking a p-value histogram. Now, in fact, you don't need to eyeball histograms to estimate control the false discovery rate. There's an elegant method by Benjamin and Hochberg from 1995 that essentially does what I showed you before, but in a more elegant manner. So the algorithm sorts the p-values. So the p-values distribution is actually shown by the points that make up the black line. So it's just the plot of the p-values by rank. And what you then do is you draw a line with slope alpha over m into this plot and go for the last intersection between the red and the black lines. And that intersection point here then corresponds to your cutoff for rejection. So you would then reject everything to the left of it, according to the algorithm of Benjamin and Hochberg, and control your FDR at level alpha. The implementation of this is actually really simple. What I'm showing you here is the actual code of the p-adjust function for Benjamin and Hochberg in R. And you can see it's really just a very simple ordering and intersection computation of p-values. So this what this method does is takes a list of p-values as input and returns a match list of adjusted p-values, which would basically correspond to the associated FDR if you make the cutoff here. So this is very well known and widely used. The paper is in fact one of the most cited paper in science ever, and it has sort of millions. I think it has millions of citations. So we can ask what's actually right. Yeah. So the next thing is I want to put this in the context of the two groups model, and in fact we can nicely understand the algorithm of Benjamin Hochberg also in the context of the two groups model. This is just to recall basically in mathematical terms already what I mentioned about the two groups model, and how the distribution of p-values is here modeled as a mixture of two components. And the main point to make here is that we can now relate the two groups model and the Benjamin Hochberg procedure. And this is done here. So basically we try to estimate its parameters from the data in a way that is sort of written down here. So this was in fact described by Story and Efron. So if we estimate our two groups model from data in the way that is described with the blue bullet points, so we just set pi naught conservatively to 1, which is an upper limit. We estimate the distribution function by the empirical CDF, and we basically plug these into the expression above for the local FDR, sorry, for the Bayesian FDR, and then choose the t that maximizes this value, and reject everything that and reject all the hypotheses with smaller p-values in this t star. Then basically this procedure controls the Bayesian FDR, and the method is in fact equivalent to the algorithm of Benjamin Hochberg. Yeah. So what we have here is a Bayesian interpretation or model, or a Bayesian interpretation of the Benjamin Hochberg procedure. Now, of course, I should say that Benjamin Hochberg procedure is purely a frequentist. There's no Bayesian model or any model underlying it. It just works on p-values, and it's an algorithm that computes something on p-values, and it can simply be illustrated or sort of visualized in this way. But I mean, as these authors that I mentioned here have remarked, it is remarkable how this procedure controls the frequentist FDR, even if the model doesn't hold, even if all the alternative distributions are different and the HI are deterministic. So that's the link between the two groups model and the Benjamin Hochberg procedure, which is very useful for what follows. And the main point now is that can we actually do better than what we have had until now, talked about until now? And the main point to notice here is that the Benjamin Hochberg method and also the two groups model only talk about the p-value from each test and treat all the tests otherwise as exchangeable. So we only have the p-values, and everything else is forgotten about our data. But in fact, we often do have interesting covariates. So for instance, in RNA-seq, ChIP-seq, CLIP-seq, and so on, we also have the normalized, for instance, the normalized means of counts for each gene, and sort of intuitive that genes with high counts have more power than genes with very low counts. In the same way, in an EQTL analysis, maybe an association between a SNP and a peak in the chromatin is more, or an expression is more plausible if it's a local association than if it's very distal and far away one, and so on. So we often have these additional pieces of information that may inform us about the power of detecting something, or the probability of something that we don't actually use if you only look at the p-values. Now, people have of course noticed this long time ago. Bioinformaticians have used these additional pieces of information when they made selections, for instance, of gene lists. And there's been all sorts of awkward combination or proposals of combining p-values with additional information in the past. I mean, one of the systematic efforts, well, one sort of 10-year-old realization was that you can in fact do this independent filtering, so that if you have, for instance, an RNA-seq experiment, have measured tens of 20,000 genes, but half of them only have really, really small counts, you may as well drop them from your analysis before even doing the tests, or in fact, independent of doing the tests. You can just drop them and just focus your analysis in the multiple testing adjustment on the remaining ones. And as long as your criterion for dropping or filtering is independent of the maldistribution of the p-values, this is a legitimate thing to do. So that's what's described in this paper that I'm citing here, and which is sort of a predecessor of what follows. But in fact, we don't need to have just a simple yes/no filtering, because in fact, the situation is a little bit more gradual. And this is sort of illustrated here with these histograms. So these are p-value histograms now, stratified from the RNA-seq experiment that I talked about earlier, that I mentioned earlier, stratified by average read count. So at the bottom right, in sort of plot 8, you see the really highly expressed genes, or the genes with the really high counts on average across all the samples. In the top left plot, you see the genes with really low counts, low average counts across all samples. And you can see that the two-component model really is different in those different histograms, or that you have evidence now that something is going on that probably corresponds to differential power between those different bins of genes. That you have more power to see something in bin 8 than in bin 1. And we want to use that information. So that motivates using the conditional two groups model. So basically it's all as before, but we have an additional variable x that we use to condition our model. So now the pi naught is not a global parameter. It's not a global parameter. It's in fact something that depends on this covariate x in our hypothesis space. And also the same way, our alternative distribution, so the fourth line in this model, is now also dependent on xi. So we are now allowing for different alternative distributions depending on the value of the covariate. So this is now exactly what we sort of want. And we can now use this. So now that we have the conditional two groups model, this directly corresponds to the weighted Benjamini-Hochberg method. So this is a simple modification of the method of Benjamini-Hochberg, where in addition we have weights for each hypothesis, so positive numbers W that average to 1. And we can define sort of weighted p-values as indicated in the second line here on this slide by dividing each original p-value by this weight. So what this means, if a weight is large for certain hypotheses, the p dash becomes smaller. If a weight is small, p dash becomes larger. So this sort of this transforms the p-values. And what Genovese and collaborators have shown already a while ago, if you now apply Benjamini-Hochberg to those modified p-values, you still have proven type 1 error control. So this is done using deterministic weights. So if the W's now come from are actually previously known somehow and are there and are deterministic, under those conditions, their usefulness has already been shown by Genovese and Röder and Wasserman. Because the problem is how to actually know the weights, where do we get them? And there's, of course, multiple different answers to this, but wouldn't it be nice if we could actually estimate them from our data at hand themselves? A little bit in the way that we have these plots where we see that probably we should weight the hypothesis from bin 8 higher than weight the hypothesis from bin 1 here. So couldn't we just use that information to get at our weights? That's the idea. And this really now leads to the central slide of this part of the talk. So that's the basic idea of independent hypothesis weighting. So for now, we have this covariate X, which indicates, as I mentioned, either, well, our covariate X that we can use to model the differences in power or prior probability. We now model this dependence by a step function, so we can basically stratify test into bins. You could think of modifications of this that also uses smooth functions, or I mean other more interesting kinds of regression. That's sort of an area of active work, and other proposals besides IW also have already used other, for instance, smooth functions for F. But for the time being, we're okay with using step functions here for F. So basically, which means to stratify test into G bins. We pick a certain alpha, split the hypothesis into K disjoint folds, and I come back to what these folds are. But you can think of basically as random folds that sort of in a very similar way as you do cross-validation and split your training samples into different cross-validation folds. But here we split the hypothesis. And now for each fold, we apply the weighted Benjamin-Hofbach procedure with a weight vector chosen to maximize the number of projections at level alpha for the hypothesis outside the fold. So we basically learn the weight vector for our different bins on out of fold hypothesis, but then apply it to the within fold hypothesis. That's the basic idea of IW. And if we now, the mathematical details of this, and also the proofs for the FDR control, both in finite sample and asymptotic settings, are in this archive manuscript that I'm citing at the bottom. I think it's also in Yingyi's mail. Go ahead.
So may I ask a clarification? So
Sure.
here you have two ways to divide your hypothesis into categories, where one is the G bins, and that is based on a covariate. The other is K folds, and that is random division.
Mm-hmm. That's correct.
So yeah, so when you do the K fold, did you do stratified division? So basically within each bin, you divide them into equal sizes K folds, and then you pull them together, or the K fold was not stratified, it's just random division?
In practice, I mean, with a large number, I mean, if you're not talking about tens of millions of hypotheses,
that's
the basically equivalent. I mean, and I mean,
right.
we're thinking about cases when both the K and the G are sort of single-digit numbers, so let's say 10 or something in that order, but we have billions of hypotheses.
Okay.
So it's not. I mean, that's not really an issue. I think both would work equally well.
Mm. Okay, thank you.
So the splitting, so random is the default in some cases, and I can come back to this. It can also be good to use explicit folds. Let's say what we did in the EQTL data is that the folds are different chromosomes. The idea that that breaks any dependencies. So if we, for instance, learn the weights for chromosome 1 for the data from all the other chromosomes, We don't have to worry about linkage disequilibrium and correlation introduced that way. So sometimes, I mean, we have independence assumptions in the proofs, and sometimes those independences, which are necessary between the different faults. And sometimes if you know something about your hypothesis, you can actually engineer your faults, the K, in a way to make them maximally independent. And otherwise, if you don't know anything, or if you don't worry about independence anyway, you could just use random splitting. Right?
And my other question is, why would you want to discretize the covariate into G bins without treating or fitting the weight as a function of the covariate directly?
And that's simply technical laziness, if you wish. I mean, fitting a step function is sort of simple. I mean, basically you have to deal with bins. If you want to fit a smooth function, you have to deal with smoothness and sort of, I mean, conceptually it would be nicer to do the continuous fit, but then I guess you're also more dependent on the specific application scenario, because X could be any domain. I mean, sometimes it's a distance, it can be average expression levels. I mean, so the data type of X can be very different. And then sort of what the best smooth function is will then depend a lot on the application example. And I guess binning always works. So there's like it's sort of the baseline. But I agree with you that it's interesting to also consider smooth functions or regression trees, or I mean, you could have a machine learning or complex regression model here that sort of fits your F. You could have a deep neural network fitting your F eventually, and I think this is also future work. For now, we just assume that X can be separated into different bins. Yeah, but there's certainly more work to be done in fitting more interesting or more complex Fs.
Thank you.
All right, I'm also almost done. Just a few more sort of results slides on using this. So what you here see in this slide is now the application to this RNA-seq example that I mentioned at the beginning. It shows you the number of the left is the number of discoveries as a function of p-value cutoff. And as you can see with Benjamin Hochberg, you, for instance, at a cutoff of 10%, you get something like 350 rejections, whereas with IHW you would get something like 600 rejections. So it just shows you the increase in power. I think in this case, maybe the increase in the number of rejections may seem inconsequential, because in both cases you have lots of genes that are rejected. In other instances, it could be more critical that sort of the differences between finding nothing at all and finding something. So if you're in a power limited scenario, which is, for instance, often the case when in the EQTL example, which I'm showing later. Also interesting is the weight function. So this, in fact, shows you the weight function that was found by IHW on the right-hand side, and what it sort of gives a little bit the idea of the independent filtering. So for hypothesis for genes with low average levels, to the very left, the weights basically become 0. And that's sort of consistent with the idea that you just shouldn't test any genes that have very low expression levels because they have no power anyway. And then there's also this interesting behavior at the right-hand side, that this sort of this weight budgeting. And in fact, in some sense, the algorithm finds it more effective to put a little bit more type 1 budget into the intermediate hypothesis, let's say into the swing hypothesis, into the swing bins, and less type 1 budget into those bins where the power is large anyway. Sort of the main takeaway is that the ranking of hypotheses now is no longer monotonous in the raw p-values. So I think the implementation of p-adjust and R, maybe in general by the algorithm, have led to this idea of adjusted p-values, so there's a one-to-one relationship between raw p-value and adjusted p-value. But this is really an artifact of a specific algorithm. And if you do more interesting of the Benjamin Hochberg algorithm, if you do a more interesting multiple testing adjustment like IHW, but also like other other proposals, the relationship between raw p-value and adjusted p-value is not homogeneous. It's not monotonous anymore. In fact, in this case, it's monotonous within each of the groups. So here you have an instance with 22 different bins. This corresponds to a situation with G equal 22. Within each bin, of course, the relationship still is monotonous. But then between bins, it's very different. So for instance, a raw p-value of 0.04 can correspond to quite a different range of adjusted p-values depending on which bin you're in, right? And then finally, this is not the histone QTL example, so the Mike Snyder data. So again, you see here, we do find a big improvement by using the distance between the SNP and the histone peak as a covariate, as we would expect. Of course, local associations are a priori more plausible and also more frequent. And we here see a comparison of power between the sort of DERMA application of Benjamin-Hochberg, then doing independent filtering using different cutoffs, which of course you would have to choose manually or sort of know which cutoff to use, and then IHW, which automatically finds this weight function, which is shown on the right-hand side. So on the right-hand side, you see the weights as a function of genomic distance. In fact, it does now 5 curves corresponds to using those 5 different folds. So as importantly, those 5 curves should fall on top of each other. Of course, in the finite sample, the finite data scenario, there's still some fluctuations between the different curves, but they have the same basic behavior of having very high weights for small distances and then falling off. And then, for instance, above about a megabase of distance, the weight is very small. So the association would need to be really strong in order to be detected. Maybe one more sort of in this direction is that the decision boundaries in such a multiple testing setup are now no longer straight lines. So if you look at the log of the p-value, log 10 of the p-value, for instance, and the covariate, if you use Benjamin-Hochberg, it just knows p-value, so the decision boundary is a straight line, the dashed red line. IHW knows also about the covariate, and it can sort of engineer other decision boundaries, like the blue one, that are maybe more adapted to the actual distribution of the 2-component model, and in this way is able to eke out more power at the same type 1 error, the same FDR. To summarize here, multiple testing is not a problem, it's actually an opportunity. There is almost all applications in biology and elsewhere, heterogeneity across tests. Informative covariates are usually apparent to the domain scientists, but they have to be independent of test statistics other than null. And I gave you some examples, like the mean counts of the gene across all samples in RNA-seq, or the distance between SNP and eQTL, and so on. But they have to be informative on pi1 or f-alt, and this is very easily diagnosed if you, for instance, do these stratified p-value histograms that I showed you. We see that the histogram of p-values empirically are different for different values of the covariate. And one way to deal with this is IHW, the data-driven weighting. So it gives you improved power without overfitting, and thus controls type 1 error. And this is basically achieved by this cross-weighting, or by this division into folds that I mentioned. And the implementation scales well into millions of hypotheses, which is also good, obviously good for applications. The package is available in Bioconductor, and you can give it a try. There's also a vignette that should be quite easy to read, that gives you sort of an idea of how to apply it to data. And as I said, there's a more sort of applied exposition of this in the Nature Methods paper, and a more formal mathematical one in the archive paper, which has been revised and hopefully at some point will also be in the journal. I'd like to thank my research group, and in particular Nikos Ignatiadis for his work on this, and you for your attention.
Thank you very much, Dr. Huber, for the talk. So now is the time for questions from the audience. So any questions? Who want to break the ice to speak first?
Yeah, I have a question.
Sure.
So this might be kind of a naive question, but overall, we would expect the weights and the raw p-values to have some sort of anti-correlation there. And so you're kind of inflating the p-values with these weights. Is then there some sort of like multiple testing going on there as well? And do you have to correct for that?
I mean, what can be going on, what I mentioned, is overfitting, that you would, in fact, if you didn't do this cross-weighting, the splitting into fold that I mentioned. So, I mean, naive approaches here would not have the splitting into folds, and it would simply sort of optimize the weights to maximize the number of hypotheses on the set of hypotheses. And if you do that, you do indeed have the problem in some situations, especially in all null situations. You totally overfit and lose your type 1 error control.
Okay.
So if I understand correctly, I mean this hypothesis splitting, which is in a way sort of more or less similar to cross validation in learning, avoids the problem of overfitting.
Thanks.
So following up with Russell's question, I think I do have a question about how did you exactly find the weights? So how did you solve the optimization problem here numerically?
I mean, in the end, the weights, I mean, G is a number of groups here. So
Right.
it's, let's say, a 10-dimensional vector, and it's a simple, I mean, in IHW, it's implemented using integer linear program. And then it's simply an optimization that runs for a little while and then gives you the result.
So you basically research with the constraint that is sum up to 1, the weights.
Yes, I mean, you don't. I mean, it's not. I mean, it's an optimization, so it doesn't do a full grid. I mean, you could do conceptually complete a full grid search, but I mean, just say computation time, you basically use an optimization algorithm. And there's still, I think there's still also now. I mean, so what we have done so far, this is described in the Nature Methods paper, is this integer linear problem solver. So we can actually, I mean, one of the things we can engineer is to make the problem convex, so that we know that we do get a global optimum. So there you have some freedom in the choice of your functions that you use. It's also a little bit comes back to your question, why do we use the step function f? I mean, if you use a more complicated class of functions to optimize over, you need also an optimizer that is able to deal with that. But in the end, I mean, it basically becomes machine learning. No? I mean, finding the best regression function for something is well, it's either a regression problem or if you use that language, a machine learning problem.
I see. And also, I also have a question about the choice of K. So what is the guidance here? The number of folds?
I think it's mostly uncritical, so we often use 5, for instance. The main thing is, I mean, as I mentioned, to avoid this problem of overfitting, you basically want that what you learn what you learn in fold K is independent, so that there's no information leak between the hypothesis on which you learn to the ones on which you apply the weights. So between the folds, so basically you want is independence between the folds. This is trivial if you have global independence of all hypotheses. If your hypotheses do in fact have some independent structure, let's say like in the genetic data you have linkage disequilibrium, then you would like the folds, for instance, to be different chromosomes, or otherwise, I mean, sets that are quite far from each other. You see, or maybe if you do image analysis, and you would have maybe dependencies between neighboring voxels, your folds might probably be different volumes that are far apart from each other.
Mm-hmm. I see. So basically, you want to make the independence, you want to make the folds approximately independent of each other.
Exactly. Yes.
And another question I have is regarding when the FDR control still holds. So that will be relying on the weighted BH procedure, right? So the requirement for the weighted BH procedure to work to control the FDR, is it that the weights must be constant and they are not really so the weights cannot be random, right? They must be constant themselves.
No, that's the point. The weights can be random here. They are random here. So the deterministic weights, that's what the Röder and Wasserman paper showed.
Wasserman, I see.
And the whole point, so with the deterministic weights, this has basically been out there for a while, and with the random weights of what we have here, I mean that's the main advantage, that we can use data-driven weights or random weights if you wish.
So here, if the weights are random, then the requirement is that I guess the weights must be independent of the p-values.
That's correct. I mean, well, the covariate. So you learn the weights from the covariate, and therefore we have these independence relations requirements. So in the easiest case, everything is independent, and then and you only need independence under the null. Yeah. So obviously. Yeah, so what you do need is that the covariate is independent.
Data you use to calculate the p-values, I think.
Say again?
So I mean that you have the data, right? You use some variables to calculate the p-value, and then you have some other covariates which were not used in the p-value calculation.
Correct. Yes. So I mean, one instance is in a t-test between two groups, you would use the t-statistics to compute the p-value, and you could use the overall mean value of all measurements or the overall variance as a covariate, or in fact the sign of the difference. So all of these covariates are actually independent of the t-statistic and the p-value under the null, but they might give you information about the prior probability. But with the sign, I mean, you may have situations in which changes in one direction are more likely in another direction. And you could learn this by using the sign of the difference as a covariate. Because under the null, that's independent of the t-statistic.
So I guess the application would be like if the p-value calculation is based on some standard statistical test, but that test cannot really use other informative covariates you have, then your strategy would be a simple way to combine the covariates with those simple p-values. And I also have a question about the first part regarding the multi-omics factor analysis. So over there, you said that you can, so let's say you have 4 views, like one is methylation, one is RNA-seq. So you can actually see how well each view, the raw data of each view can predict a factor. In your case, that was factor 4. And you use that to say, okay, how to explain my factor. So I wonder whether we can just look at the loading matrix you have. So for each factor, you have the loadings for each view. Can we just use the loading, the magnitude of the loadings to say, okay, how much my factor is related to this view?
Exactly, yes.
Okay. And also, I wonder what is the difference between the MOFA and simple concatenation? Let's say that I have those features 39, 5,000, 5,350. What if I just concatenate them and then do PCA?
It's a good question. So one thing is the sparsity, that you can introduce, so we can have
missing values.
The structured sparsity in here.
Okay.
And the other thing is how, like, the influence of the different variables. So for instance, in the example I'm showing here, we have 39 mutations and 5,000 gene expression values. If they concatenate, it may well be that the 5,000 just drown out the signal from the 39. And in this multiple factor analysis, you have a better chance of weighting that against each other. But I agree. I mean, it's this in the end and really depends on how you structure your priors and how you do the estimation.
I see. Thank you.
I mean, basically in the model, you for instance, here we have room for different epsilon m's, so that the noise term can be different for different views.
Okay.
And if you quickly concatenate, that would probably typically not be the case, right?
Yeah. PCA basically assumes Gaussian noise. Yeah. If you just do the naive
and
version.
even, I mean, since it's the least squares, it also sort of gives each feature the same weight. So
Okay. Any more questions from the audience?
I have a question.
Yes.
So
who is speaking? Could you speak up a little bit? Your voice is a little low.
Oh, in your weighted procedure, you divide data into k subsets, and for each subset you learn the weights using the other data. Then it sounds like you apply the weighted BH procedure k times. In my previous research long ago, we found out that if we divide data into subsets and apply BH procedure just to the subsets separately, we tend to inflate the errors. I wonder if you would have some level of inflation.
no, I mean, this is the whole driving of this project. I mean, why we even talk about it, that we can now do this without inflating the type 1 error. I mean, I agree. If you do like in the sort of obvious or naive approach, you have the problem of that you may overfit, that the weights then may start fitting fluctuations in your observed p-values, and in this way you can then get really bad violations of type 1 error control or of FDR control. And the whole point of this work is to show that you can avoid this, essentially with the idea of the cross weighting.
Okay. Well,
yeah.
Certainly an alternative approach would be just wait until at the end when you learn all the weight and then do a single weighted BH.
Oh, but it doesn't matter.
Okay.
Does it?
I guess that will be the issue. If you learn the weights from the same data and then you apply the weighted BH, then I think that's the overfitting issue Dr. Huber was referring to. If the weights and the BH was applied to the same data, I think in his procedure, the key is that by using the cross-validation, the weights were learned from some data, but when he make the discoveries, it's from the other data that were not using that were not used to get the weights, right?
Oh, well, I guess I understand that for each for the k subset, each subset you learn g values, and then at the end of the day you have a k times g values.
Yes.
Yeah, for the combinations of the bins and the flows. So my alternative suggestion would be just keep those k times g values and then apply the BH procedure in a single step.
That's a good question. I need to check. My intuition is that it doesn't make a big difference, but it would definitely be interesting to check how those two options compare. I mean, applying the procedure separately for each code. I mean, as it's written down here, or as you say, save all the weights and then apply the procedure once to all the weights.
Right. I have another question. So here, certainly the research is driven by a covariate that's potentially informative but not used in p-value calculation. So you want to incorporate that into the weighting. Mm-hmm. I wonder if this procedure, even though you don't have that covariate, it's just this learning procedure to me is very interesting. What happens if there's no covariate? You just randomly split data into G bins and also K folds independently, and then learn the weights. Would that help?
I mean, it shouldn't. I think if the covariate is not informative, the weight should be flat.
Okay.
And then you would basically be thrown back to the normal Benjamin Open procedure.
Okay. Thanks.
Thank you very much for the question and Dr. Huber's answer. And do we have any more questions from the audience before we conclude? This is a very rare opportunity. We're doing this cross-continental communication.
Oh, yes. I have a question for the first part of MOFA. I remember you mentioned that we have K latent components. So yeah, still how to choose K? Is it determined by the data or it's just a predefined?
This is a very good question, and I think it's in the end it's similar as in PCA decomposition or in NMF. I mean, it's a model complexity parameter where we currently have heuristics to pick it, but there's no good theory.
Okay.
I mean, usually we just use a large number, I mean, a large enough number, and then look at the factors and see whether they make any sense. I mean, as an example, before we had 7 factors, but only 3 in the end really we interpreted.
Okay, I see. And yeah, just
I mean, it's an exploration technique. It's not an inference. It's sort of it's good. It's good if it helps people understand the data. It's not. It's a first step. It's not a. I mean, MOFA should be the first step in analysis, followed by many other things. It shouldn't be the last step.
I see. Yeah, and the additional question about obviously the components you find out by MOFA. In your later example, you show that factor 4 is kind of informative, and I just wonder what happens to the first 3 factors.
What about the other factors? Is that the question?
Oh, I mean, I remember there is a slide. You find that factor 4 is kind of related to the biological meaning. Yeah, I wonder what happens to the first 3 factors.
Okay, yes. So the first 2 factors, F1 and F2, are in fact related to sort of well-known biomarkers or tumor properties. So this IGHV and trisomy 12 are sort of clinically important properties of these tumors that distinguish their biology and also their outcomes. So I think those 2 factors are quite interpretable. The third factor, F3, it seems to be exclusive to the mRNA view, as you can see here with in the heat map. I mean, here it's all white and it's just dark in the 4th pixel here. And we think it's probably a batch effect. There's something to do with maybe with the sequencing and the order in which the samples were run and whatnot.
I see. Thank you.
Okay, any further questions? We have a lot of students here, so I would like to encourage our students to ask questions. Even if it's some questions about the background, I think they're still very welcome.
I have a basic question if there's still time. So regarding the hypothesis, if I have two pairs of hypotheses that are in different bins, and the covariates were used to create the bins, if I'm understanding correctly, is the assumption that these two hypotheses are conditionally independent given the covariates?
What do you mean by a conditional by independence of hypothesis? I mean, one thing we assume is that the null distribution, that the p-value under the null, so is independent of the covariate, both within the hypothesis and between pairs of hypotheses. So the p-value of one hypothesis should not depend on the covariate of another in a different bin.
I see. I guess
yeah.
I think it was touched upon earlier. You mentioned like linkage disequilibrium and this type of thing.
Such, yeah.
Okay, thank you.
I mean, obviously, yes, exactly. So I mean, in such cases when you have linkage disequilibrium, so neighboring SNPs. So the hypothesis that originate from neighboring SNPs, let's say the association with the expression value, would be highly correlated.
I see. And so the covariates are unique to each hypothesis?
That's correct. Yeah.
And
right,
so Gabriel, yeah, so I think I can add one more thing. I think, yeah, people have shown later since the BH procedure was published that if you have positive correlations among the hypotheses, then the BH procedure can still control the FDR, even though you will lose some power.
I see.
Yeah.
It's actually really hard to break it. I mean, I agree with Jingyi. There's a lot of worrying about independence between hypotheses, but it's actually really hard to construct counterexamples where you can break the type 1 error control through correlations.
Yeah.
And the other thing is, I mean, if correlations between hypotheses are a real big problem, then maybe multiple hypothesis testing is the right approach anyway. I mean, you should probably first go to a dimension reduction or some other kind of multivariate model. I think the fact that you do multiple testing, screen multiple hypotheses, already implies that in some level you believe that they are independent.
Mm.
Like in a drug screen, I mean, or otherwise, or for instance, or let's say if you have a brain imaging application, maybe similar. Maybe you go through every voxel and test for something. It's probably true that neighboring voxels are correlated, but you would assume that at least for voxels that are far enough apart, then you're independent. And so it doesn't make sense to screen through all of them to look for effects.
Yeah.
But if you have a global correlation between everything, you probably don't want to do this multiple hypothesis testing anyway. You want to do something else.
I see. Yeah, it makes sense. Thank you.
Thank you. And any further questions?
Hi, Dr. Huber. I have a very naive question. So for your independent hypothesis weighting procedure, I wonder if there's any easy way to check if the independent assumption is held here. Like, for example, your p-values are independent with your covariate. I remember that you use like a mean count or mean log count for your DSIC2 example. But yeah, I guess this might be kind of intuitive for biologists, but I still wonder if you have some suggestions on how to check this assumption. Thanks. Yeah.
No, I mean, this you can mathematically prove. There's no need for intuition. I mean, in that case, you can simply prove explicitly that the test is constructed. I mean, for instance, in the DESIC, the test is constructed so that under the null, the p-value is independent of the mean. Same in the t-test.
Yeah.
I mean, that's the way the tests are. The tests are constructed to have this property.
Yeah. I guess this is the case. But how about like other potential covariates, like, sorry, I may not have a good example here, but thanks, yeah.
No, I mean, obviously, if if you use a, so I'm just trying to find the table. So this is, of course, the place where domain knowledge goes in. So here I have some examples of covariates where you can basically show either by mathematics directly on the tests or by domain knowledge that there's independence. For instance, in the GWAS again, the minor allele frequency, the tests are constructed in a way that this independence holds, that this is a sort of nuisance variable, in the test. People are sometimes tempted maybe to use covariates that are in fact correlated with the p-value, but then in fact you would be really in trouble, because then you sort of have that double dipping, and really you would invalidate the validity of your results. But all of these examples that I'm showing you here, by construction, the p-values are independent of those covariates other than null. Let's say sample size is another example that, so for instance, if you have many different tests, but they have different sample sizes, again, the test for each sample size, for each n, the test is constructed such that the p-value, null distribution is uniform, independent of what the sample size is. Yeah.
All right, thank you very much for the questions and the answers. And yeah, I will just ask for the last time, do we still have more questions from the audience? Please don't be shy. If you do have questions, just unmute yourself and ask. Don't need to wait for me to ask you to ask questions. And I think the details are in the papers we sent in the abstract. So if you want to check out further details of the contents, please read the papers. I think that would be the best place to get many answers, I think.
I'm also happy to take your emails if the question comes up later. It happens to me a lot that the question I only really come up with afterwards, thinking about it, and and then I'm happy to, of course, take your emails as well.
Yeah, that would be great. Thank you so much. So if there are no more questions, let's thank Dr. Huber for his talk and for giving this virtual seminar to us across the Atlantic in Germany. That's really special for us. Thank you so much.
Thank you. Have a great day.
Have a good day. Have a good evening, actually, over there.
Yes. Thank you.
Thank you. Thank you. Bye-bye. Bye. Bye.